Greetings. It is with great pleasure that I welcome you all to the first lecture of our course, Introductory Animal Physiology. In this course, we will delve into the fascinating world of animal physiology, where we aim to understand how various physiological systems work together to maintain life and adapt to different challenges. Today's lecture serves as an introduction to animal physiology and homeostasis, the foundation upon which all other topics will build. The study of animal physiology not only helps us comprehend the marvels of living organisms but also builds the foundation for applied sciences, veterinary medicine, and animal husbandry. As we embark on this journey, I encourage you to actively participate, ask questions. Your curiosity and enthusiasm will be the driving force of our learning environment. Hi, myself Dr. Tulasi Raman, who is Assistant Professor of Animal Physiology, Department of Veterinary Physiology and Biochemistry from Faculty of Veterinary Animal Sciences, Institute of Agricultural Sciences, Banaras Hindu University. Thank you for choosing this course. I look forward to guiding you through the intricacies of animal physiology and ensuring that you come away with a comprehensive understanding of this critical subject. Let us begin our exciting journey with Lecture 1, Introductory Animal Physiology and Homeostasis. So, before entering into the physiology subject, let us see some history related with the discoveries and then the important events related with the physiology. You know that Hippocrates, he is the person who is called as the father of physiology or medicine. And the next important, the forefather of the physiology, you can say, in the history wise, Aristotle. He proposed the humoral theory of disease. The humoral theory of disease consists of four components. One is hot, cold, wet, and dry. So, if you see the components, the earth, or you can call it as a black bile, then water, which is equivalent to the phlegm. Phlegm is nothing but the mucus which is secreted by the respiratory tract, but this is not the mucus secreted by your nostrils. Okay, so you can say it is a sputum also. Then air, which is equivalent to the blood, and fire, which is equivalent to the yellow bile. Now, the next person is that Claudius Galen, who is very, very important in the physiology. He is the first person who used experiments, hence he is called as a father of experimental physiology. The humoral imbalances can be located in the specific organ, such as sanguine is related with the blood, and yellow bile means it is choleric, and then phlegmatic means it is related with the phlegm, mucus, and then black bile means it is equivalent to the melancholy. So, he proposed the human body consists of three connected systems, Claudius Galen, such as brain and nerves, heart and arteries, and then liver and vein. So these are the three connected systems by which the homeostasis is maintained by the body was proposed by the Claudius Galen, and he is called as a father of experimental physiology. In addition to him, there are many discoveries which are taken place in the field of physiology or medicine where the Nobel laureates and Nobel prizes were given. So, among them are the Jean Fernel, who is a French physician, introduced the term physiology. Okay, and then Joseph Lister, he also studies the homeostasis as well as inflammation. Then Von Haller, he is an important person. He contributed to the study of nerve and the muscular physiology. And then Pavlov, as you all know, he contributed, his work is understanding the behavioral and then the physiological responses was a huge contribution for the field of the physiology, Pavlov's. And then Alan Hodgkin and then Sir Andrew Huxley. So they explained the nerve impulse propagation, which is another important milestone in the discovery or in the field of physiology. Then Crick, Francis Crick, and then James Watson. You might be well know about this names of the Crick and Watson, who discovered the DNA, right? And then Rosalind Franklin, his work is related with the, it's a molecular physiology. So there are many discoveries are there related with the physiology. Of course, I have highlighted very few. So if you want to know the complete list, you can check it in the Nobel Prize website. From there, you can able to know what are the different discoveries happened and in for which discovery and when they have announced the Nobel Prize and all, and who are the contributors. So what is physiology? The physiology is nothing but study of the basic functionalities of a living being, or you can say animals. So animal means it is animal physiology, and human physiology means it is the basic functionals of the humans. So that is human physiology. So it is a study of regulation of change within the organisms and the science of body functions. So anatomy is related with the study of the structures, and then physiology is nothing but study of the functions, normal functions of the body. So this is you can define the physiology in that way. Then coming to the levels to understand the organization, there are different levels are there to understand the organization. So the first and foremost, the basic thing is an atom. So atom is nothing but it is the smallest unit of an element. Now what this smallest unit of an element atom does, this atom also is made up of some protons, neutrons and electrons. Okay, so these atoms will combine together and form a molecules. Many a times in the molecules what happens, sometimes an electron will go, or either it will accept or it will remove any of the electrons from the shell. So as a result it will become an ion. Okay, now these molecules all together it forms the macromolecules. So the macromolecules, when we say, you can call it as a carbohydrate, protein or the lipids and vitamins and minerals. And these macromolecules are grouped together and form organelles. So organelles, where it used to be present, it is present in the cell, right? So these organelles are grouped together and form a cell, and then the cell are grouped together and form a tissue, and these tissues are grouped together and form an organ, and organs are grouped together and form an organ system, and these organ systems are grouped together and form an organization or the organism. So this is the level of the understanding of an organization in an animal or a humans. So coming to the, in other ways you can tell the level of organization in a body is that the chemical level, cellular level, tissue level, organ level, system level and organism level. So the same way I have given in a different way, where the chemical aspect means only the atom, molecules and the macromolecules are coming together, whereas in the cellular aspect if you see the organelles and the cells, and these are all comes under the cellular part, and tissue, then ultimately tissue will become organ, organ become organ system become the organism. So these are the levels to understand a organization. Now in this introduction I am going to give you some of the ideas as well as some of the key concepts which is required to understand the different mechanisms or for the journey of animal physiology. Now the systems of the body, there are various systems of the body are present in the body. The first is that integumentary, that means skin, and then the skeletal system, muscular system, then nervous system, then endocrine system, immune system, cardiovascular, respiratory, digestive, urinary and reproductive. These are the major systems are present in the body. In addition to that, you can say the lactation system also in animals, which is of very importance. Then the next is that homeostasis. So far you have seen the important findings which is related with the physiology or the advancements related with the physiology, and then we define the physiology, then different levels of organization. Now we'll move on to homeostasis. So what is meant by homeostasis? All those functions what you are going to study, these all systems and the all mechanisms are going to achieve only one thing, that is homeostasis, nothing else. So what is that homeostasis? Homeostasis means it is same. Homeo means it is same or similar, and status means it is the stand or stay. Okay, so that means it is the maintenance of the internal environment, okay, at stable. That is the definition of a homeostasis. What it is? A maintenance of the internal stable environment. where irrespective of the different sensory information or inputs from the internal environment or from the external environment. Irrespective of that, if a body can able to maintain a stable internal environment, then this will be called as a homeostasis. Like for example, whether it is a winter or summer, our body is maintaining the temperature of 37 degrees Celsius. So that means it is trying to maintain the homeostasis irrespective of the external factors or the internal factors. Then the W.B. Cannon is use the term homeostasis first, and the Claude Bernard is the one who described the term homeostasis. It is one important objectives you can say in terms of physiology. And then how these homeostasis are maintained. These homeostasis are maintained or regulated by many factors. So the first factor is that internal environment, and then the next external environment. What are those internal environment? The internal environment such as the concentration of nutrients, then gaseous concentration, then different waste products, then the pH, the water concentration, salt concentration, and the electrolytes, followed by the volume and pressure, and then temperature. These are some of the internal factors which helps or which regulates the homeostasis. Regulating means it will either increase or decrease some mechanism by stimulating it, right? Then the external environment. So the external environment means other than these factors, which is in terms of internal. If you take away all those factors which from the internal side, then all those will belong to the external environment. So next will be the how these factors which helps the body, which regulates the homeostatic mechanisms by which a control systems are there. So the control system, how this control system will help to maintain the homeostasis? It helps to maintain the homeostasis by detecting the deviations. So first of all, the body is at 37 degrees Celsius. Suddenly, if it is fluctuated to 39 degrees Celsius, that means there is a deviation from the normal body temperature. So that should be identified by the factors or the control system. Once it is identified, the next aspect is that it has to be integrated this information with other relevant information. Is it only the temperature issue, or is there any volume issue, or any temperature or any pressure issue? So that has to be, all the informations must be collected and it has to be integrated. Once it is integrated, then it will be sent to the higher center where the appropriate adjustments are made to restore the abnormality to the normal range. So this is the function of a control system. And how this control system, why it is working? It is working just to maintain the homeostasis, that's all, for maintaining the stable internal environment, remembers. Now these control systems are broadly classified into intrinsic or local, and the second one is extrinsic or systemic. The first one we have seen regulating factor, where the internal and external we discussed, that is environment, whereas here the control system type, it is extrinsic and intrinsic. Extrinsic means it is systemic in nature and intrinsic means local in nature. So intrinsic means it is within the cell or within the organ, you can say, whereas extrinsic means it is just, it is away from the organs, or it is outside of the organ, you can say, that is systemic, right? So example for this intrinsic or local mechanisms, you can say that there is a person who is doing some strenuous exercise. So what will happen? The person's oxygen level, their requirement is increased. So as a result, the blood which supplies the capillaries, which is supplying blood to that particular muscle, right? The muscle will give a signal by increasing the concentration of carbon dioxide. It will give signal to the capillaries, okay? And the capillaries will respond to that. How? It will just causes the vasodilatation to that particular muscle so that it will supply more blood. As a result, the oxygen requirement will be fulfilled. So this type of adjustments is called as local adjustment. That means what? Something is required locally, so that information will be given to the nearby locations, areas where the temporary modifications can be done. As a result, the problem will be solved at locally. So this is the perfect example of the intrinsic or local mechanism. The next is that extrinsic or systemic mechanism. So in that, the many systems are involved to correct any issue like acid-base imbalances or any type of mechanisms, RAS mechanisms, then hypovolemia, hypertension, all those will be corrected by extrinsic or systemic mechanisms. Now, these extrinsic or systemic mechanisms are broadly classified into three types. One is called as the positive feedback, then negative feedback, and then the third one is feed forward feedback or the mechanism. Okay, so that can be three types of feedback mechanisms, you can say. The first one is positive feedback, second is the negative feedback. These are the most common feedbacks you will find across the books, and the third one is the feed forward mechanisms. So the positive feedback means that a component is increased in the blood, it will give a stimulus feedback to the either the target, I mean the hypothalamus or pituitary, or to the any higher center, where it causes further increase the release of the particular component or chemicals or a hormone. Then that mechanism is called as positive feedback mechanism. One example is that the oxytocin release. The oxytocin release during parturition. So parturition means it is delivery of a baby, you can say. In animals, this is called as parturition, delivery of a calf or any animal by the mother. So during this parturition, the head of the fetus will try to come out. So as a result, it stimulates. So it will stimulate the neurohypophysis. So neurohypophysis will release the oxytocin, and this oxytocin will come, and in turns it causes the relaxation of the muscles, and further release of the oxytocin it promotes. So that means what? Oxytocin itself is sufficient to increase the concentration of the oxytocin, or even the fetus pushing will give the feedback so that it increases the oxytocin. Again, the increased oxytocin and the pushing of the fetus, and then again it goes on. So the oxytocin's function is to causes the contraction of the uterine muscles. So as the oxytocin come and binds with the receptors, the uterus get contracted. So when it contracts, what happens? The fetus will try to come out. Again, it stimulates the uterine muscles, and it's sending the signal to the neurohypophysis, where again it releases the oxytocin. Then again oxytocin will come, and then again it will act on the receptors, and it causes the myocontraction. As a result, again it will be pushed, then the same thing. That means it is keep on increasing, right? One event will causes the increased release of the another event. So this is called as positive feedback mechanism. Next is that negative feedback mechanism. Negative feedback mechanism you will find in many places in the body. So the components of the negative feedback mechanism, such as there is a deviation in a controlled variable that will be detected by the sensors. Once it is sensed, then the information should be sent to the higher centers, that is called as integrator. Once it reaches the integrator, then integrator will give a command to the effectors. Okay, now these effectors in turn it send the response through the motor neuron, and ultimately it will try to bring the variable to the normal range. So these are the components of a negative feedback mechanism. Now let us take it in terms of some example, right? So first is that hypothalamus. Hypothalamus, what it does, it causes the releasing hormones, and then the releasing hormones will go and act on the anterior pituitary. Then the anterior pituitary releases the tropic hormones. Then the tropic hormones acts on the target organ. Then the target organ releases the hormones, and this hormone will produce the biological effect. This is the general outflow. Now what happens? The negative feedbacks, there are many types of negative feedbacks are there. One is long feedback loop, next short feedback loop, and then ultra short feedback loop. So the long feedback loop, as you know that the hormone is the final product. So the hormone, okay, when its concentration increases in the blood, then automatically it sends signals to the hypothalamus, okay, the master gland, where it causes decreasing the release of the releasing hormones. So this is a long feedback loop. That means from the product to the origin, right? The next is that short feedback loop. The short feedback loop, what happens when the concentration increases in the blood, then it sends signals to the anterior pituitary level only. That means one step low, where it controls the tropic hormone release. Okay. Now in the earlier step, what happens? Decreasing in the releasing hormone, that itself come and act on sufficient to cause the decreased release of the tropic hormones. But in the short feedback loop, it will directly acts on the anterior pituitary and causes the decreased release of the tropic hormones. Now in the third category, that is ultra short loop or the feedback loop, if you see, the tropic hormone concentration when it increases in the plasma or in the serum, then that is sufficient to causes the decreased release of the tropic hormones from the anterior pituitary. This is the ultra short loop mechanisms or the loop. So these are the different loops you can say. Example is the T3, T4 hormone releases. So these and all. Then the feed forward mechanism. The feed forward mechanism means it is working based on the anticipation. Okay. So anticipation means when there is a food or the feed in the stomach, then it is knowing that there is a requirement of a higher glucose concentration will be expected in the plasma. So as a result, it stimulates the insulin release so that insulin will be ready for the whatever the glucose are present, so that will get absorbed. So how it is working? The first is that it's a feed or the food which is in the stomach, it will get digested. When it get digested, it liberates glucose. So the glucose concentration increases, then the glucose will in turn causes the release of the insulin from the beta cells. So the insulin will come and increases the absorption of the glucose into the cells, right? And the beta cells anticipates that further increase of the glucose will takes place. So it anticipating that one, and then it causes the increased release of the insulin further. So as a result, whatsoever the macromolecules get digested and it releases the glucose, it will get absorbed, so that the maximum absorption rate can be reached by the body. Next aspect is that fluids. So the fluids can be broadly classified into extracellular fluid and the intracellular fluid. Now why it is called as a fluids? It is not water. The fluids is just because it contains some solutes in it. Whereas in the volute water, you may not find the solutes in it. So that is why in the body, whatever the water components are there, that will be called as a fluids, not as a water. So cerebrospinal fluid, right, aqueous humor, vitreous humor, all those synovial fluid, everything is a fluid only, not a water. But the maximum 98 to 99% it is made up of only water molecule. Okay. Now the total body water, I told it is broadly classified into intracellular fluid and then extracellular fluid. The intracellular fluid is almost formed 2 to 3 proportion, whereas the extracellular fluid it is of 1/3 proportion, the composition. If you take the extracellular fluid, in that the 15% is of interstitial fluid. The interstitial fluid such as lymph, CSF, synovial fluid, aqueous humor, vitreous humor. In addition to that, 5% is of plasma volume. Okay. And then the transcellular fluids such as intraocular fluid, CSF, digestive tract fluid, synovial, urine, bile, these are all called as a interstitial or the transcellular fluids. That means between in the cells. Now the next aspect is that in the introduction, the proteins. You know, the proteins are the building blocks of any organism or the organization. Okay. So the proteins, all the physiological changes is mediated in the body by the proteins only, the maximum. And so these proteins play a major role in different ways. So according to that, based on that, the proteins are functionally classified as catalysis, reaction coupling, transporters, structure and signaling molecules. So catalysis means what? It increases the ability of the chemical reaction rate without altering its equilibrium. So that is called as catalysis. The next is that reaction coupling. Reaction coupling means there are two types of reaction: spontaneous reaction, non-spontaneous reaction. Example is that one downhill and then one uphill reactions. So downhill example, you can say if you are driving a car, you will fill the petrol. So if you want to move forward, you have to, the fuel has to be spent, right? It will burn. and as the fuel concentration or the volume is decreasing, on the other hand you are moving forward or you are taking the uphill, or that's what is called as move downhill reaction and the uphill reaction. So that is a reaction coupling mechanism. Then the transporters, there are many transporters are in the body, and then the structure and signaling, it is a building block of the body, and then the signaling molecules such as chemokines, neurokines, and many type of signaling molecules it releases when there is an issue. And the protein functions is based on the binding sites as well as the ligand, that lock and key mechanisms. And the another important aspect of a protein is that allosteric property of a protein. So by which the four mechanisms the allosteric property of the protein is achieved. One is that it will bind, the lock and key mechanism. Then second is that sequence specific ligand binding. You might have seen in the genomics and all, it is the nucleotides ATC, right? So it will bind with the complementary pairs only, right? The same way, that is sequence specific ligand binding. Then the third is that electrical field that is surrounds the protein. Based on that, the voltage changes, it will respond. So that will be called as a voltage gated channels. Okay, so these channels will be responsible or respond to that electrical fields that surrounds that particular channel or the membranes. Then in response to the mechanical force also, the allosteric property takes place, and that is achieved with the help of an example is that mechanosensitive proteins, which is example you can say in the vestibular apparatus. In the vestibular apparatus you can find the mechanically gated potassium channels are present. So this is respond in relation with the mechanical force but not with the voltage gated or any other or ligand. Okay. So what is this allostery? Allostery, whenever this type of mechanisms happens, the conformational change of the protein will take place. As a result, it triggers the downstream process and leads to the biological effects. Next will be the biomembranes, the cellular membranes. You can say it is not permeable. You know that because for many components, and the charged particles, example ions, then polar molecules, which is a molecular weight greater than 100 daltons. So these are all not permeable, whereas permeable, if you take crucially important polar molecules such as urea or water or the lipid soluble molecules and all those gases, so these are permeable easily. And then the movement of the molecules usually takes place based on the concentration of the solutes in the fluid, and this is strongly affected by the different forces such as the concentration as well as the pressure, which forms a chemical potential, and then the voltage, which contributes for the electrical potential. So these forces are called as a free air forces, and that will act as a source of free energies. Then once the electrochemical potential is generated, that is the free energy of the substance from all the sources per mole of the substance, then it will be called as an electrochemical potential. Then there will be a spontaneous transport to occur when the difference in the electrochemical potential is required in between the places. So when it reaches the equilibrium, then the movement of the ions will be stopped. Otherwise, the driving force is sufficient to bring the chemical potential as well as the electrical potential. Next will be the transporters. The materials move spontaneously from higher extracellular concentration to the lower extracellular concentration. This type of transport is called as a diffusion or passive transport, where there is no requirement of energy because it is moving in the favor of the concentration gradient. So until and unless it reaches the equilibrium. Then the next is that facilitated diffusion, where the channels as well as the carrier proteins are involved. Without the involvement of ATP, then it will be called as facilitated diffusion. Then the another transport is called as active transport. So far we have seen the simple transport, right? And then the next is that active transport, that is a passive transport we have seen, now active transport. So active transport means involvement of ATPase is required, that means it is moving against the equilibrium. And then these active transports are broadly classified into primary active transport and the secondary active transport. In some books you will find the tertiary active transport also. In primary active transport, it spends the energy, where secondary active transport, it utilizes the energy of the primary transport and then it moves, and the tertiary transport, it uses the substrates from the secondary transport and then it moves the molecules. Then the next transport is the co-transport or counter-transport. Co-transport means it moves together, counter-transport means it goes against. And then symphotus again it is moving together, and then antipotus it is against, that is a counter-transport you can say. Then unipotus, only one molecule will be moved from inside to the outside cell or outside to the inside cell. Then the next important definitions are Solute, solvent and osmosis. You know, solute is nothing but when the physical property of any molecule changes, then it will be called as a solute. When the physical form is retained, then it will be called as a solvent. And when you mix the solvent and solute, you will get the solution, right? Then osmosis. Osmosis means mostly it will move into, you can define it in terms of either solute or in terms of solvent. So in terms of solute, if you take, it will move from the higher concentration to the lower concentration. That means the solute concentration, if you take, if there is a higher concentration is present, then the water will try to move from the lower concentration solute to the higher concentration solute. Okay, so that is how you can define the osmosis. Then specific gravity, it is the ratio of weight of volume of blood or urine to the water. So that is how you can define the specific gravity. Then osmotic pressure, you can then colloidal osmotic pressure or the oncotic pressure. Then osmolar and the osmolar, when the number of particles or the solutes expressed in terms of 1 kg, then it will be called as osmolar. Or the number of particles or the solutes which is present in 1 liter, then it will be called as osmolar solution. Then the channels or carriers can be leaky or ligand gated or voltage gated. Then the other type of transporters are paracellular and the transcellular. Paracellular means it is side of the cells, transcellular means it is through the cell. Endocytosis, it is the engulfing, and then exocytosis means it is splitting of the cell. So today we have seen the history, what are the different contributions in the field of physiology, and then we seen introduction, then levels to understand organization, then homeostasis, different feedback loop system, then what different types of fluids, then proteins and its importance, and then biomembranes and transport mechanisms. Of course, I have not discussed the cell because it is one of the basic information that you know, that all the functions of the different cell organelles. So that's why I didn't cover. With that, I am ending the today's lecture. Thank you.